The actual biology is quite important. It's important to understand a bit about the biology of colorectal cancer from a screening point of view. So I'm sure most of you will understand a bit about cancer. Does everybody understand that cancer is a disease of the cells, where the cells become uncontrolled and they start dividing and spreading in an uncontrolled way? Is everybody aware of that? Okay. So this is what obviously happens in bowel cancer. So the lining, or the mucosa, or the inside of the large bowel, the cells, and we'll come on to the reason why in a moment, but the cells start to lose the usual controls that keep them in place and keep them ordered. So they start to sort of heap up in the bowel. At the early stage, this is not cancer, it's a precancerous condition. We call this in the large bowel an adenoma. And an adenoma is basically a benign tumour of the large bowel, which has the potential then to go on to become a cancer. So in the vast majority of cases, the cancers start as a benign adenoma, and then further changes occur in the cell, and the cells start to migrate into the bowel wall, and they start to invade into the bowel wall. They then spread to the lymph nodes, which are the glands which drain the bowel wall, and into the blood. And once they get into the blood, they can then spread throughout the body, particularly to the liver and to the lungs, but they can spread anywhere. But the liver and the lungs are the two common sites where what we call metastases end up from bowel cancer. So we've got this sequence of going from a small changes in the cells into an adenoma, into a cancer, and we call this the adenoma-carcinoma sequence. And of course the importance of that is, I think the importance of that is quite obvious. Can anybody see why clinically that's an important thing, that you have this pre-malignant condition and then a malignant condition? Did anybody get any ideas why that would be important clinically? Why it's, and particularly why it's important in screening?
Well, pick up the adenoma.
Yes. Yeah, by taking out the adenoma, you can prevent cancer. And there's now very good proof from properly conducted scientific studies in big populations that if you remove adenomas, you can prevent colorectal cancer from occurring. And therefore it follows, if you can detect adenomas, you can actually prevent colorectal cancer from occurring by removing them. I'm going to say quite a bit about prevention, but a bit later on, because I think that's a very fundamental issue for all of us, is how you prevent colorectal cancer. But one way of doing it is to detect adenomas and to remove them.
Are adenomas what you normally call polyps?
Yeah, yes. The word polyp and adenoma can be used interchangeably, but it's not actually correct, because a polyp, all adenomas are polyps, as it were.
Not all types.
But not all polyps are not adenomas. So you can get polyps which don't turn into cancer. Nasal polyps, for example, but you even get polyps in the colon that don't turn into cancer. We've got a question over here.
Is there a typical timescale associated with that progression?
Yeah, I think it's all kind of guesswork, because nobody has done the experiment of leaving adenomas and seeing what happens to them. Or in fact, there was some work done a long time ago, but of course now that we're convinced that they do turn into cancers, it would be unethical to leave adenomas to see what happens. But we believe that the average timescale from the first changes to developing invasive cancer is between 10 and 15 years, probably. But that comes from pretty imperfect evidence, and we're not going to get any really robust evidence from that now.